Okay, we'll start with some questions where we left off already. Sorry about that. Pause. Pause. So now we are going to look at what happens in prophase 1, and this is where things kind of get interesting. All the stuff that we saw happening in prophase in mitosis, all that stuff is going on. So all those prophase activities are happening now. And what we need to look at is what makes this different. So early on in prophase 1, we have those homologous chromosomes. Remember that each of those chromosomes is consisting of 2 sister chromatid. So we've got 4 DNA strands, 4 chromosomal strands, and these join together to synapse, and they form a tetrad. 1, 2, 3, 4. This process of synapsis is the pairing of the homologous chromosomes that occurs during meiosis. The ends usually are attached to a nuclear envelope, and the synaptonemal complex that we talked about a little bit earlier forms, getting us a nice tight fit between the two sets. And each thing is matched up gene by gene, locus by locus. Everything matches up perfectly. So where these things are bound, and we see this perfect alignment, we also see those, remember, we have those cohesin proteins that we talked about in mitosis. Those are found at the centromere. So when synapsis occurs, the two homologs, this is one and this is the second, join up in that synaptonemal complex. And that tetrad now consists of the 2 homologous chromosomes, each having 2 sister chromatid. So this, remember, is a single chromosome. It has 2 strands. These 2 strands are the sister chromatid, and they are exactly the same, right? This is its homolog over here. So we've got the homologous chromosomes. These are the sister chromatid of this chromosome. When we start looking at non-sister chromatid, and this is important because this is where we get some of the more interesting stuff that happens in prophase 1 going on. So the non-sister chromatid, same chromosome number, but coding for, if it's eye color, one's blue, one's green. So that's what's going on with the non-sister chromatid. Coding for the same things, but slightly different. Here, if we're looking at eye color here, blue eyes and blue eyes. Here, green eyes and green eyes. But the non-sister chromatid, blue eyes and green eyes. So coding for the same thing in the same exact position, but slightly different. So this is where we see crossing over happening. And crossing over occurs between those non-sister chromatid. That's why I was kind of making a big deal out of it. We're still looking at the homologous chromosomes, and this occurs at this chiasmata. And this is where the 2 homologs, the 2 homologous chromosomes, remain in contact, and we see crossing over occurring. And the idea of crossing over occurring is basically we get an exchange of genetic material. So if we had blue eyes down here, green eyes here, green eyes here, there's an exchange that takes place. So now this one still has its blue eyes, but now this homolog, or this rather sister chromatid, has green eyes. And the same thing here. This one originally had green eyes, but now that crossing over has occurred, this sister chromatid now has blue eyes. So we literally get an exchange of genetic materials. And this is one of the reasons why we have to be really careful about how everything lines up perfectly, because we want to make sure that we're exchanging gene for gene. And this is another way that we get genetic variation. As you might assume, because all of this stuff is going on, prophase 1 lasts longer than it did in mitosis. Now, in prometaphase 1, we're going to see similar things that happened in prometaphase in mitosis also. What we want to remember is that while the same things are going on, the microtubules are attaching to the kinetochore, they're attaching to something that's become fused because these two sister chromatid are not going to separate yet. So these kinetochores are actually joined together. So the microtubules are attaching to that single kinetochore, or that joined, I should say, kinetochore. Everything that happened in prometaphase in mitosis is going to start happening here. So all the same activities. We get the kinetochores forming, they fuse together on the sister chromatid. All the prometaphase activities are happening. And then in metaphase, this is where we see the tetrads. So this is where we get the homologous chromosomes are lined up, and we get those tetrads. That's where that crossing over occurred that we talked about. Now, we talk about the orientation of each pair of homologs on the spindle as random. Basically, we've got these color-coded to make it a little bit easier. So here we can see the red on this side, the blue on this side, but it could line up so the red is on this side, the blue is on this side, but look here, this is where it's different. So it could line up so this is still red and blue, but now over here, this is blue and red. And this is for a cell that has very few genes. So the more chromosomes any cell has, the more complex this gets. So where does it land, one way or the other? And this is where we get this idea of independent assortment. The orientation, the fact that it's random, it's not set up that this is always going to be the red side and this is always going to be the blue side. It's completely random. It could happen either way. This is independent assortment. And that is the end of meiosis 2. Now, one of the things that we want to get back to is this idea of great genetic diversity. So with only 2 pair of chromosomes, we look at this idea of here's a 2 pair, we've got 2, and this is the 2 pair. 2 squared is 4, so we have 4 possible genetic combinations. And these are the different possibilities. Now humans have 23 pairs, so now we're looking at 2 raised to the 23 power. So this exponent is how many pair of chromosomes. When you do that math, it's something over 8 million. So that's 8 million possible combinations just from independent assortment. So that's pretty significant. And then when we get into crossing over and we see genetic exchanges on those non-sister chromatid, we get even more genetic diversity. So as we move on to anaphase 1, remember prometaphase 1, same things are going to happen, lining up at metaphase 1. And then anaphase 1, this is where we've really got to be paying attention again, because what happens here is we see the homologous chromosomes separated. The sister chromatid are still joined together. Now remember, in mitosis, in anaphase, the sister chromatid are the things that are separating. Here, sister chromatids stay together, but the homologous chromosomes separate. So now we go from 46 chromosomes to 23. In telophase 1, those spindle fibers and homologous chromosomes are working to move to the opposite poles, and we end up with that haploid number of chromosomes. What happens during the next portion is sort of dependent on the organism. We may see a variety of things happen. We're not going to worry too much about what's going on in interphase here. We know cytokinesis is going to occur about the same time as telophase most of the time. But the end result is we've got these two haploid daughter cells. And those two haploid daughter cells, that means that in humans, each cell has 23 chromosomes. It's just that each of those chromosomes has two sister chromatid. So that's the important difference here. So interkinesis, again, variable, but we never see synthesis again. So that's the big deal. This is when the cell now moves into prophase 2. And what's sort of interesting here is as we move into prophase 2, we're going to start seeing a lot more similarities. So let's look at this question. And we pause. 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 So now let's get into prophase 2 or meiosis 2. So remember the chromosomes didn't replicate, so we still have this haploid cell. Each of the chromosomes, remember again, consisting of two sister chromatid that are still joined at that centromere. Everything that we did at the end of meiosis 2, if the chromosomes uncoiled, if the nuclear membrane started to reform, any of that stuff that happened is going to go the reverse again. So we need to recondense chromosomes. We need to break down anything that reformed. This isn't hugely common, but it is one of those things that we do want to make sure that we mention. But then again, the chromosomes are basically going to start moving to the metaphase plate. All the stuff is going to be basically the same that happened previously. We're going to see those spindle fibers appearing, all those prophase activities. Then we see all those really prometaphase activities. The spindle fibers are going to attach to the kinetochores of those sister chromosomes. Almost exactly the same thing that happened in mitosis, except it's a haploid cell instead of a diploid cell. And then when we go to metaphase 2, the chromosomes again lining up at the metaphase plate, exactly the same as they did in mitosis. Same exact process, except we're going from haploid to diploid. And this is now at anaphase 2, where we split those sister chromatid from each other. And now those chromosomes are going to move to the poles, just like anaphase in mitosis. At the end of all this, the daughter cells are still haploid. We still have 23 chromosomes in each of these cells. The sister chromatid have been split. Same generic process that happened in telophase for mitosis, except, of course, the fact that it's now haploid instead of diploid. But we see the nuclei reforming, all those things that went away come back. Cytokinesis is occurring. But now at the end, we've got these 4 haploid daughter cells. And this means we're either going to have 4 sperm or 1 ovum and 3 polar bodies or bar bodies, depending on who you read. This is one of those things that I'm just going to tell you to read through and stop if you want to. As a review for you, this is just another recap. And this is another recap. I actually took this out of your book. It's a little icky. in the slide, but I do have the page number on your from your textbook. And remember, you can still get that textbook. You can download the PDF for free.